It's my pleasure today to welcome you to this new webinar from Scilife, and on behalf of Scilife, I thank you very much for participating in our training. Today we are going to discuss a hot topic in QC labs. The topic is how to handle out of specification results, out of trend results, and out of expectation results in labs. Okay. My name is Angel Buendia, and I'm the knowledge manager at Scilife. And the training is divided in two parts for those who's the first time you're joining these trainings. The training itself takes 45 minutes, and we have with us Stephane Lieven. He will provide the training. I will present him later on, all right? The training will be followed by a Q&A session of about 10 minutes at the end. So this is a 1 hour training. So what is the training about? Okay, so if you are from QC, you know that QC personnel play a major role in ensuring that the products they test meet the specifications. So these people are fundamental to ensuring the quality of products and also patient safety as well, okay? And the webinar provides an overview of insights and strategies that are needed to handle 3 topics: OOSs, OOTs, and OOEs, that give me a major concerns on final analytical results in QC labs, okay? So after the training, we'll have a Q&A session where you can ask anything you'd like to know about Stephane's presentation. So feel free, as commented, to post your questions using the Q&A button. And today, Stephane will answer as many as he can, okay? If we don't have time, don't worry, because we are going to prepare a PDF document with the most important highlights of this training, as well as the Q&A session, all the questions and all the answers, all right? So please allow me to introduce Stephane Lieven, Senior Life Sciences Consultant at QBD. So hello, Stephane, good morning, and thank you very much for being here today. And
hello, good morning, Angel.
Go ahead. Yeah, please go ahead with your training.
Okay, thank you very much, Angel. Good morning, everyone, and thank you for joining the training on handling of OOS and OT and OE in laboratories results. My name is Stephane Lieven. I'm a chemist, and I work in pharmaceutical industries for more than 20 years. I have a background in QC analytical testing for more than 10 years as a technician in QC. Afterwards, I work for more than 8 or 9 years as supervisor in a QC laboratory. So I face a lot of OOS and OOT and so on during my career. And since 7 years, I work as consultant, and now I'm consultant for QBD, Quality by Design, in validation, QC activities, analytical validation, and method transfer. Okay, so we can start the training. Today I will try to explain to you all tips and all what the regulatory are saying about these, and we will deep dive into how to process when we are facing OOS, OT on OE results. And at the end, I will try to give you some tips and tricks to be proactive in a goal to avoiding this kind of situations. Okay, so let's start with some definitions. to be sure that we are talking about the same topic and what does it concern. So OOS is abbreviation for out of specification results. And what does it concern? It concerns all test results that fall outside the specification or acceptance criteria established in each drug application or drug master file, official compendia methods, or by manufacturer in-house methods or procedures. The term also applies to all in-process laboratory tests that are outside of established specifications. So these definitions comes directly from regulations. So there are different regulations, European, US, ICH, and so on, but they are all in line on the topics, and definitions are quite the same in each regulations. We are also talking about OOT, which means out of trend results. What is an out of trend? An out of trend is a result that does not follow the expected trends, either in comparison with other batches or with respect to previous result collecting, whatever during a stability study or starting materials and in-process sample testing, or during analysis of semi-finished or finished final or finished products. When we talk about OOT, that doesn't means that these results are necessary out of specification. They can be in the specification, but don't look like typical data point as usual. So it should be considered for environmental trend analysis, such as for viable and non-viable data. And it can be controlled with trend analysis with the action limit and warning limits. But we will talk about this little bit later in the training. And we are also talking about OOE, out of expectation results. These are quite different as from the OOT results, because an out of expectation result is a completely atypical, aberrant, anomalous results that doesn't have any relation with the weighting results. So that are results that are still within specification, but are unexpected, questionable, irregular, deviant, or abnormal. For example, if we take an example of chromatography analysis, you could have normally a chromatogram that shows, for example, two active ingredient peak, and for unknown reason, at that moment, you will see a third peak on the chromatograms. So this is characterized by as not of expectation result, because we are just attending only two peaks on this chromatogram. Okay, these were for the definitions. It's important to know that in the training, as in the guidelines, we are almost focused on OOS specification results, and OOT and OOE are less discussed, but the process is exactly the same than the OOS results. So when I told about some rules regarding OOS results, it also concerns OOT and OOE generally. Okay, so definitions are done. Then what's in the scope of the training and the topic? So all batch release testing and testing of starting material, as we are performing in QC laboratory, for example. All the in-process control testing that are used for batch calculation, batch release, and decision, and if in a file or on a certificate of analysis that will be reported. Also, the stability studies with markets or commercial destination are concerned, and finished product and or active ingredient, pharmaceutical ongoing, and follow-up of stabilities. The stressed stabilities are not concerned. It's quite normal because in stressed conditions, we are forcing the degradation of the product, and then we are expecting to find some out of trend or out of specification results. So this is not in the concern topic. Also concerned are the previous released batch used as a reference batch sample in an OOS investigation that show an OOS or suspect results, and the batches for clinical trials. So to summarize the situation, whenever we are in a GMP environment, almost all results are concerns from this topic, because in GMP conditions, whenever we are generating results, we cannot disregard these results without any good reason. So we have to document all the things and follow the basic rule of the GMPs. Okay, then let's go a deep dive in the regulatory insights, and let's see what they are saying to be compliant to this regulation and the GMPs. Okay, so we've got, of course, the US FDA, Food and Drugs Administration guidance for industry. They have posted a special guidance related on this topic, and called Investigating Out of Specification Test Result for Pharmaceutical Production. And last revision was done in May 2002 and 20. There is quite same guidance for the British part of Medicinal and Healthcare Product Regulatory Agency, MHRA. And their guidance is from 2013 about out of specification, out of trend investigation. Sorry, the last update is not in 2013, but in 2018. We've got also the ICH, which have also a document on this topic. It's on the ICH Q7, and on the chapter 11, part 1, there is a specific topic on quality control sample, and analysis of results generated. So then comes inevitably the OOS, OT, and OE results. Okay, so yeah, that's what I previously already said in the beginning of this presentation, but it's important to remind it that although recommendations are intended for OOS results, the same investigation principle may be applied to out of trend and out of expectation results. And that the FDA regulations require that an investigation has to be conducted whenever an OOS test result is obtained. So we cannot dismiss this investigation and disregard results without documented correctly all that have been occurred in the lab. Okay, so what are the best practice in laboratories to be ready to treat this kind of investigation? Then I will explain to you what is the process that is recommended, highly recommended, required by the regulations. So this is a general flow chart, and we will go deep dive in detail on each phase of this flow chart, but it's a good view before to start. So the process is that we will perform testing in laboratory. We will find results. If this result is not OOS, okay, no problem, we can report the results. If we are facing no OOS results or OT or E, we will open that's what we call the phase 1 investigation, and this phase 1 is related to the laboratory itself. So we will investigate if the results is coming from a mistake from the lab or not. That will be the first phase. If this investigation concluded that there is no lab error, we will go to the phase 2 OOS investigation, which is called the full-scale investigation. And then in this investigation, we will go through other process. So the production, manufacturing will be involved, and all associated process outside of the lab. And if in this case, if a manufacturing production error is identified, it has to be corrected if it's still possible, and if not, the batch has to be released as non-conform, or has to be documented and discussed with the qualified people, the qualified person, to decide on the conclusion of this batch. If no error, if an error is detected in the lab previously, then is the same process, documented and correct the error in the lab, and report new results, or in different case, retest first sample and report the results. But we will explain the process deeper a little bit later. Also, aside from these investigation, are related CAPA that will be open. So CAPA means corrective and preventive actions. That means documentation and questionable about the specific case of each OOS, OOT, OOE encounter, to be sure that all corrective action for the batch concern have been taken, and to be sure that preventive action to avoid recurrency of the same case of, for example, error in the lab will be reworked. Okay, that was for the flowchart and general view of how to proceed. Then we can go deeper in the principle in the investigation process. The purpose of the investigation, and it's really important to keep always this in mind, because what is the purpose of the investigation is to determine the cause of the OOS results. We have to know why we face OOS results. It's really necessary to avoid recurrence and to grow up in your society and in your lab, and avoid recurrence and solve the problem. We cannot disregard any results without any good reason. So for this, we have to determine the root cause of the OOS results. So the source of the OOS results should be identified either as an aberration of the measurement process or an aberration of the manufacturing process. Even if the batch is rejected based on OOS results, the investigation is necessary to determine if the result is associated with other batches of the same product or other products. And the batch rejection doesn't negate the need to perform the investigation. The FDA regulation part 211 required that written record of the investigation has to be made, including the conclusion and the follow-up of the activities against this OOS, even if the batch has been rejected. Okay, to put all these process in place, the basic is to get in place a procedure, and all the people involved that are well trained to these procedures, and this procedure has to explain all the process that will be followed, and should be divided in 2 phases to reflect that the OOS results can be caused by either laboratory error or product fails to specification. So as mentioned earlier, according to phase 1 investigation and phase 2 investigation. Investigation OOS test results, including the responsibilities of the laboratory personnel, is really important. The laboratory phase of investigation, additional testings that may be necessary when to expand the investigation outside the laboratory, and the final evaluation of all test results. So it's really important to involve as soon as possible the laboratory technicians, because that's in the lab that the OOS occurs, and the first actor to be the right persons to solve the situation are the technical laboratory technicians, because they know exactly what occurred, and they have perhaps observed some things which is not as usual in routine testing and so on. So I really encourage all people to go deep dive in the lab and discuss with all people concerned, all different laboratory, if there are testing performed in, for example, 2 or 3 different teams, and involve all the people around the table to discuss about topics. It will be really easier to find a solution. And a really important thing to note is that I say that procedure should be well in place. So it is the first thing. And it means also that if there will be during the investigation retesting of samples, reinjection of some solution, or so on, resampling before retesting, it has to be first documented in a report of investigation before going to the testing, because test into compliant is not permitted, really not permitted. We will come again on this this information a little bit later, but just keep it in mind. So the phase 1 laboratory investigation, okay, so should include an initial assessment of the accuracy of the laboratory's data. Whenever possible, this should be done before test preparation are discarded, including the composite or homogeneous source of the alicot testing. Why to do this? This is that by this way, hypotheses regarding laboratory error or instrument malfunction can be tested using same test preparation. For example, if you are injecting sample on chromatographs, and during the investigation, you remark that there is a bubble in the injector, for example, but it should be good reason that results are not accurate. And then if you have kept all the samples, you could perhaps re-inject for the investigation, not for the final results, but for the investigation, the same preparation, and prove that the bubble in the injector was the responsible of the OOS generated. And then if this is the case, you can go in the investigation and to confirm that it's not a real OOS, but a wrong OOS due to a technical problem, and then it can be solved. But if you don't have any more all the tests prepared during the initial testing, it becomes really more complicated to get the proof of the root cause. So just always keep test preparation and all reagent and concerned material before a test sample has been released in general. I know it's not always easy in practical, but it's really, really, really recommended. Then if this initial assessment indicates that no meaningful errors were made in the analytical method used to generate the data, a full OOS investigation should be conducted. So that means that if we, during the first investigation, lab we don't find a root cause, we have to go deep dive for full scale investigation, and that will involve other department, contact manufacturer, producer, and so on. So here are some topics that should be discussed and investigated during the lab investigation, phase 1. So, which I called analyst responsibilities, but these are, at my sense, responsibilities of every people work in a lab, is to be sure before I start testing, I must be sure that my instruments are qualified. So I have to check if my balance is qualified, my HPLC is qualified, and so on and so on. I must check if instrument function programmation have been correctly done. So just verify properly if this is the case. Verify if the method step has been followed correctly. Don't to avoid mistake of dilution of sample, for example. Be aware, for example, when you face some results with factor 5, 10 times, 20 times, all these rounds make we think that we face problem of dilution. Take a glassware of 10 milliliters instead of 20 milliliters, for example. Also, during testing, QC technicians, laboratory analysts has to be trained really good on GMP practice, and encourage the technician to report any observation during testing, even if at the time of the testing it seems to be okay. This time my flask and my solution is quite pale yellow color, and normally it should be orange, but it's not a criteria of the requested testing. So okay, it doesn't matter, but yeah, record this this information, but perhaps it can be an information important in case of OOT or E or OOS results, and if it's not recorded at the time of testing, afterwards we don't remind it, or it's quite complicated. Okay, and also always retain the sample and inform your supervisor about the situation as quickly as possible, to avoid losing time in the investigation, because some samples have a degradation time so short. So the shorter we know, we can start the investigation, the easiest way we can find solution. So these are only a few topics, because there are a lot of topics, but there are the main topic to investigate first in the laboratory investigation. And also in parallel, the supervisor or the management team or team leader and so on has to be involved in the process, and will go in the lab, as I already mentioned it earlier, to discuss with the analysts about what occurred and so on, and what are the color of solution and the injection and so They will check together the system suitability and method specification, review all the data, standard, reagent use, preparation of the solution, the dilutions, check if everything are correct, verify if the sample tested are first the right sample to be tested, to see on the package if this is the good formulation, if they have not changed the molecular structure, for example, and that it was not communicated by the manufacturer. It's just an example. And then the formula of the calculation should be perhaps different. Is the sample stored in a good flask container in good conditions? You will also to check all the calculations and if the method meets validation criteria. Be sure that the analysts that perform the testing are really well trained to the methods, and fully document the assessment lab phase in the report of the OOS procedure. So all topics that are mentioned here and in the previous slides should be discussed, but furthermore documented in your OOS report. That's required in the regulation and during the investigation process. So that, for example, 5 years later, you are not present in the lab, you are not working there, technicians have been changed. But when we have to discuss again about the topic related to 5 years ago occurred, we can open the OS report and just have all the documentation, and we can retrieve all the information required. That's the basic of the GMP required. Okay, then when we finished the phase 1 laboratory investigation, we have to conclude about this investigation. And first, there is an obvious lab error that is identified. That then I should say it's the best case. Then we will document in the lab event, lab report, OOS report, whatever it has been called in the procedure, the results as non-conformity results. And in this case, because it is linked to an obvious error of laboratory, the results could be invalidated, and we can restart testing on a new sample or on the same sample, depending on the case, or just recalculated the results, also on depending of the result of the investigation. So it's really important that invalidate results only when the investigation shows the OOS is due to a clear assignable cause, not in other cases. It's really important. And if we are thinking that it is due to a specific cause, but we are not sure, then we are mandatory to go to phase 2 investigation. Okay, then In the case we are going to phase 2 laboratory investigation, this may consist of full-scale extensive investigation out of the laboratory, and also involving production process review or a laboratory review. Then the objective of such an investigation should be identify the root cause of the OOS results and take appropriate corrective and preventive action on the specific situation. So a full-scale investigation should include a review of the production and sampling procedure and will often include additional laboratory testing. Sorry, just small technical problem with the sun. Then written record of the review should include at minimum the following information. So when you finish, you are in the investigation, you have to document a clear statement of the reason of the investigation. So summarize the situation. A summary of the aspect of the manufacturing process that may have caused the problem. The result of a documentation review with the assignment of the actual and probable cause. The result of a review made to determine if the problem has occurred previously, really important, and a description of corrective action. So all these actions in accordance to be sure that we will resolve the actual situation and that we didn't face the same problem previously and that we won't face again this situation later in the future. Okay, then during this huge step of investigation, we will probably have to retest some sample and or in all cases reporting results. This has to be always in accordance with your procedures and respecting accuracy of the results and principle of integrity and documentation. Then, when we have additional laboratory testing, a full-scale OOS investigation may include additional laboratory testing. A number of practices are used during the laboratory phase of investigation, and these include retesting a portion of the original sample or resampling for retesting on a new sample. So in case it concerns a retesting, the investigation may involve retesting of a portion of the original sample. The sample used for the retesting should be taken from the same homogeneous material that was originally collected from the lot or tested and yield the OOS results. Decision to retest should be based on the objective of the testing and some scientific judgments. Then a predefined retesting plan should be documented before performing any retest or resampling is really important to avoid tests into compliance. Then in accordance to include retest performed by an analyst other than the one who performed the original test. For example, it's a strategy. A second analyst perform retests should be at least as experienced qualified in the method as the original analyst, but it can also include same analyst on another equipment if it's the equipment that is suggested to be the root cause of the OS, and all kind of topics that has been discussed previously. But always be sure that a predefined plan is really well documented before starting the retesting. And then mention again, testing to compliance is not permitted, and that the number of testing may vary depending upon the viability of the particular test method employed, but should be based on scientifically sound principle. And conclusion, repeat testing until a passing result is obtained. Then disregarding the OOS result without scientific justification is unscientific and objectionable under CGMPs. Okay, then in case of a clearly identified laboratory error, the retest result would substitute the original results, and then all the original data should be retained. However, an explanation should be recorded about the initial results that has to be discarded due to lab error identified. If no laboratory or calculation error are identified in the first test, there is no scientific reasons for invalidated initial results in favor of passing retest results. Then all test results, both passing and suspect, should be reported and considered in batch release decision. So it's really important that we cannot disregard any results without a root cause analysis well defined. In case of resampling, the goal of this investigation is to conclude that the original sample was prepared improperly or was therefore not representative of the batch quality, and has to be collected with the manufacturer and in good condition, and afterward the process will be the same as explained in the retesting process. Then we are finally coming to the reporting of test results and use of appropriate about the average or not. So in general, if the sample is considered to be homogeneous, using average can provide more accurate results and will justify some different preparation, independent preparation, and then it makes sense to make an average of the results. If you are injecting, for example, 2 times same solution, it's considered as one simple results and not individual results for each injection, because it's the same sample injected. You just test your injector for in the case of injection are made by HPLC, for example. This is a distinct difference from the analysis of different portion from a lot intended to determine viability within the lot, and from multiple full analysis of the same homogeneous sample. Some cases, for example, when we are making uniformity of content, dissolution test on just one pills, for example, the use of the average has no sense and is not appropriated because the test has to demonstrate, has the goal to demonstrate the viability of the method. So in this case, no average is taken into account. Inappropriate use of averaging. yeah, that's what I just explained concerning uniformity of dosage unit form. Okay. Okay, so furthermore, on the OS reporting, when we face outlier tests, then it means that a value may be obtained that is markedly different from the others in a series obtained using a validating method. Such a value may qualify as a statistical outlier. An outlier may result of deviation from prescribed test methods, or it may be a result of variability in the sample. In all cases, an outlier cannot be disregarded without root cause identified, and then has to be well documented in the report. So in some cases, for biological assay, there was a specific chapter due to viability of the methods. For example, the USP 111 are talking about how to treat an outlier, but in general, for validated chemicals tests where we suppose that the product is homogeneous, then outlier test statistical is only analysis of statistic testing, but in all cases, it will not identify the root cause of an extreme observation, and therefore should not be to use to invalidate suspect results. So, concluding the investigation, to conclude the investigation, the result should be evaluated, batch quality should be determined, and a release decision should be made by the QP. If a revealed cause has been identified, a suspect result is invalidated, and if not, it won't be the case, and the result will be retained. And invalidation of discrete test results may be done only upon observation documentation of test event that can reasonably be determined to have caused the OOS results. So first case, we confirm the OOS, then the batch should be rejected. We call investigate. If it's inconclusive, all results should be given for consideration in batch disposition decision, and it's up to the qualified person to decide about release or disposal of the batch. And then we arrive finally at the proactive some tips and tricks to avoid OOS in the laboratories. Then I come again on control chart that I call them maybe your saver, because if you have a look on this graphic, but just without understanding how we are making a control chart, we can already see that if each analysis is recorded on a graph and trend about the results during time, you can see that, for example, the first red cross is all about the acceptance limit, and a few analyses later there is one just beyond the limits, and just you can see some trends during testing, and that can be alert to avoid and control your process before facing some wrong OOS or OOT OOE results. Okay, so principal root cause of testing of OOS occurred during due to a lab mistake or internal error, sorry. first, the training, be sure that there is a continuous improvement of the technicians. Don't fall into routine mode, and always question yourself about the results at each time of testing. Take the time to train people, will save you time. Knowledge of your process, be aware of all the change control and the critical parameter about the method testing and the produce. Good communication may avoid a lot of problem, and also consider possible impact from change in the lab water loop works, temperature control, and so on and so on. So to conclude this training, I guess it will help you for the future. And yeah, conclusion, philosophy, quality is never an accident, but the work of a whole team. I really think it's a true sentence. And I will conclude this training by my philosophy. Then when I in the lab is think twice and test once. Okay, so thank you very much for this training. I think I take a little too much time, but we arrive to the Q&A session if we still get time. Angel?
Yeah, okay. Yeah, this is the end of the training. So we have extended a little bit, but I'm going to propose one thing to the audience. So based on the fact that we have received a lot of questions, what about if we extend the Q&A session? Stephane, do you have 10 more minutes so we can cover some of them?
Yeah, of course.
Yeah, I want to apologize for the attendees that need to leave now, but don't worry about that, but because we are going to collect all the questions and we are going to give all the answers. We will include this information into the PDF document you will receive later on by email. So if you have to leave now, don't worry about that. If not, if you want to stay some more minutes with Stephane, yeah, feel free to post your questions using the Q&A button below, right? Good. I hope that all the audience found this information very useful. Thank you, yeah, Stephane, for shedding light on OOS, OOTs, and OOEs. So let's go to the Q&A session now, right?
Right.
Oh, yeah. I'm going to ask you some questions, right, Stephane? So let's see some of the questions from the audience. Okay, one about the sponsor. This is one of the topics. There's an anonymous attendee that is asking, when should the sponsor be informed for an OOS or an OT by the contract manufacturer organization? Either when there's the initial OOS result or when the phase 1 is concluded. What do you recommend, Stephane?
I recommend to inform the manufacturer, producer, and so on as soon as possible. But if there is an obvious reason already identified, if when the OOS result is generated and you already know that it is due to a lab error, it's not mandatory to inform the customer because it's an internal error. But in all other cases, yes, I would assume to inform before starting the lab investigation phase 1 the customer. Why? Simply for first reason is that you will get some delay in reporting testing because of the investigation. But furthermore, perhaps already at this time, the customer or the producer will inform about some important information related to this OS and can perhaps help you to investigate to your investigation. So yes, I should recommend to inform as soon as possible the customer, producer, and so on.
Yeah. Okay, absolutely. Good. Yeah, I hope this answers the question. Good. Let's continue with another interesting topic is from Parasuramu. How do you establish the limits for an out of trend? Do you use 3 Sigma, 6 Sigma? What is your recommendation?
My recommendation is that it will depend on the specification of the product. Because, for example, if you analyze a raw material, starting material with a specification from 99% to 101%, for example, so the target is 100%. Of course, your acceptance and alert limit will be shorter than if you are testing a final product with a specification from 90 to 110%, for which you've got a range of 20% beside the target. So I would say that there is no clear rules, but it will depend on the product tested, and the most important is to start to record the trend at the time zero of your testing, and define the 2S or 3 Sigma can be done a little bit later on. And so particularly in the first point of the training, because the acceptance limit should not be fixed, but they will vary depending on the training of the process.
Okay, so yeah, it also depends on the outcomes of the validation characteristics of the methods, right?
Yeah, absolutely.
Good. Well, another question which is really interesting, and I know that from the auditor's point of view, you're going to have a lot of discussion, is once you cover the phase 1 investigation and you find that the investigation is not conclusive, so there is no clear root cause. So if you want to retest, how many samples from the same homogeneous samples you retest? What's the appropriate number?
I would also say that there is no defined rules about it, but if, for example, the initial results is coming from one preparation of sample, I would say that you will prepare 2 additional samples to be sure that your results are sufficient for statistic use. And it's always easier to walk on 2 legs. So if I only retest once, and I'm not sure of the results, I've got only 1 additional results, or I can be sure that that the first one is OOS and the second one is within specification, so you cannot conclude anything. So I would say that to test more samples than the initial testings should be better. So if you test initially 2 samples, I would recommend to test 4 or 6 samples to get additional results, and can say, okay, I've got 3 pair of results that are within specification, and only the initial results are out of spec. So okay, my root cause can be more assignable.
Yeah, I think also that, Stefan, the good thing is that it should be appropriate to have your procedure establishing your SOP, depending on the type of testing. For example, it should be nice either if so. I think it's better for consistency to have some guidelines,
Yes,
depending on the case, right? So you do not test it 3 times or 6 times or 7 times, depending on the day. So I think it should be preferable to write it down. I know that this gives a lot of discussion because the FDA does not establish exactly what is the appropriate number, because it depends on the analytical method. But there is an example there that for seven retests, right? And this is like a magic number. But in any case, it depends. No, it depends on the analytical.
Yeah, it depends. Method. But as I mentioned in the training, it always has to be documented in the retest plan before proceeding to testing, because to avoid if I retest some sample 2 samples and I found new results OOS and 1 non-conform, okay, I will then test again 1 sample to get 3 results conform and 1 OOS, and then it becomes test into compliance, which is really not accepted by regulation.
Yeah, sure. Good. Well, let's see. Well, we have 2 more minutes. So what about if we answer an additional question? This is going to be the last one, right? What is a general acceptance timeline for closure of an out of specification investigation?
It's a good question. It's a good question. I would say that it also has to be documented in your procedure. So in the OS procedure, the time of closure of the investigation has to be mentioned, and a general requirement, in my opinion, should be 30 days maximum for closing a full-scale investigation. And it seems to be a quite huge time, but it's not a quite huge time. If you have to repeat testing in the lab, sometimes it takes 3-4 days and time to review the testing. So that's why it's really important to start investigation at the day number 1 in the lab, to avoid delay, because we know all that delays are not good concern when we're facing OS results.
Yeah, absolutely. This is the maximum priority, is to start as soon as possible. And yeah, because at the end, the target is to find the root cause. And yeah, having the solutions prepared is very important, because as you commented, yes, Stefan, they can degrade. Sometimes stability studies for prepared solution do not stay more than 24 or 48 hours. So in just 1 or 2 days, you have to.
Yeah, absolutely. Yeah, absolutely.
Good. So, right. Thank you very much, Stefan.
You're welcome.
So we have well, I think it's the end. We have passed about 10 minutes, but I think it was really interesting to cover some of the questions. You're going to receive the rest in the following days. So I appreciate everybody for being here and for posting your questions. So you can visit our websites at Scilife and QBD. So let us know if you need anything from Stephane about this topic, or from Scilife about our electronic quality management system. So I want to thank you, everybody, for being here. I hope to see you in the next following training session. Okay. Be aware of our websites. We have very interesting topics in the following months. So thank you very much for being here, and see you another day.
Thank you very much. Bye-bye.